generation sequencing's output sequences may have errors. The likelihood of correct base assignment, that is, the quality of the nucleotide base calls, is related to the signal intensity and purity of the fluorescent signal. Possible causes of poor quality fluorescent signals are, for example, 1. Incorporation of the wrong base during PCR, which results in a strand with a different sequence. And since original and modified sequences are amplified in subsequent PCR cycles and sequenced, there will be light emissions of different colors from a single cluster at a particular sequencing cycle. 2. In a sequencing cycle, a dNTP rather than a ddNTP may attach, or there may be no reversal of termination during the washing stage, leading to light emission inconsistency with strands in the cluster where this defect has not occurred. 3. Small distances between clusters result in signal overlap. The sequence's output file shows you the base sequence quality in ASCII characters for each string of nucleotides sequenced. The quality score is derived from the formula Q equals -10 log of P, where P is the probability that the base is wrongly called. A good quality score of say 30 translates to 1 in 1,000 chance of the base call being wrong. Quality control metrics that can be obtained are, for example, the per base sequence quality plot showing the distribution of quality scores at each position in the read across all reads. For good quality sequencing output, most reads should have base call quality mainly in the green range. The per sequence quality scores plot of good quality sequences would in most reads have a high average quality score. The per sequence GC content plot showing GC distribution over all sequences should in good quality sequences have GC content normally distributed with a central peak corresponding to the expected percentage of GC for the organism studied. These and other quality metrics help inform decisions on processing to improve the quality of sequences. Processing involves trimming to remove, for example, ends with low base quality or adapters, filtering to remove, for example, reads with a certain number of unidentified bases or a certain number of bases of poor quality. Finally, assembling the processed sequences should show different reads over the same region having the same nucleotide at each location. If occasional mismatches are present, they are base calling errors, and removing reads with mismatches improves data quality.